All right, folks, I see 18 people here. 7:05, so I'm going to start. I was told that this arrangement for today was what was the most requested thing, so I'm not sure why less than half the class is here. But let's keep on keeping on. So I believe I mentioned to you all that we're starting urinary system today. And what we do today, everything we do up to this point can potentially come on your in-course test, right? So please keep that in mind. Now, you should be seeing my screen. You all should also know that next week, I believe that is when the in-course test is due. I'm having a little issue that I'm trying to sort out for next week. But come hell or high water, unless of course I get shot. As a matter of fact, even if I get shot, I still gonna make one test for you, right? So please keep that in mind. The test is coming, right? Remember, everything up to what we're about to complete today. I won't go past what we do today. Everything up to this point, right? All right. So let's start. All right, let's talk about stuff you already know. You already know what the urinary system is. You already know that it is comprised of the kidneys, ureters, and the bladder, right? Mainly. The reason I don't add the urethra necessarily to it, even though in some books they do add the urethra, is because the urethra, well, I mean, is not always primarily, well, how should I put it? In females, the urethra is only for the disposal of urine. Of course, in males, the urethra runs through the penis, which slightly complicates matters, right? So when we look at the urinary system of the female, we can confidently say, hey, urethra, right? In some books, they make things more complicated by saying, hey, the urethra is not a dedicated part of the urinary system, right? Weirdly enough, they did not label the bladder here. Anyway, so keep that in mind, right? But for my purposes, I think we should just consider urethra a part of the urinary system. Let's not make the distinction. I don't see the point of it. Okay, so that said, what is the function of a urinary system? Once again, this is something you all have done to death. So you already know the major function of the urinary system, of course, is the removal of excess fluid, minerals, some waste products like urea from the body, right? That is its main function. Yes, there's going to be reabsorption occurring there as well, right? But that is reabsorption in the context of removing fluid. And by that I mean you're taking the fluid out of the body, but while you're moving it out, you pull out what you want out of it and let the rest go, right? So the main function is still going to be the removal of substances from the body itself. Shaniki, I seen your roof for whatever reason. Now, that said, let's see it now in more scientific ways. So, we just spoke about the main function, right? As it says, it filters blood plasma, eliminates waste, returns useful chemicals to the body. It's the first one there because it's the most important. It regulates blood volume and pressure, of course, because if you're removing fluid from the blood, you must be regulating blood pressure. That's all. Especially if you have the ability to reabsorb fluid and send it back to the blood, right? Then it regulates osmolarity of body fluids. Remember, please, that osmolarity is basically, and we're just talking basically, not at all the chemistry, so you're supposed to know this, but it's basically looking at the concentration of dissolved substances, right, in your body fluids. So, of course, we're talking about sodium and chloride and potassium and calcium and all these other things. You know, there are proteins and amino acids in there as well that all affect your osmolarity. And why is osmolarity important to you? It's very simple. Osmolarity determines how water moves, right? So the osmotic concentration of the blood is going to influence whether water leaves your tissues and goes into the bloodstream, or whether water leaves the bloodstream and goes into your tissues. And when water moves, other substances move as well, right? So the osmolarity or osmotic concentration of your body fluids is very, very important to maintain osmosis, homeostasis, all of these lovely things, the balance, right? Now, we also have secretes renin and activates angiotensin and aldosterone system. This here, of course, has to do with the reabsorption of water back into the blood. Right? So we'll talk about that in some detail later, but it's simply that. This is talking about reabsorption of water, but it uses sodium as a means to reabsorb water. Now, you also have secretes erythropoietin, which controls red blood cell count. Now, if I asked you what controls the production of red blood cells, most of you will not mention the kidneys. But believe it or not, your kidneys are directly involved in the production in activating the production of red blood cells. When erythropoietin is produced and released into your bloodstream, it causes your bone marrow to generate more red blood cells, right? Now, it also regulates the PCO2 and acid balance. Y'all should know what PCO2 is by now. You should be tired of hearing me talk about PO2 and PCO2, right? Partial pressure, right? So PCO2 is looking at the concentration of carbon dioxide, essentially, in arterial blood. PO2, same thing, in arterial blood, right? Now, how does it do this? Because it can remove chemicals or compounds, I should say, that contain carbonates from your bloodstream. And remember that your bloodstream can transport carbon dioxide in the form of carbonates, right? Or can produce carbonates. So when you remove carbonates from the blood, you also remove carbon dioxide, right? That, of course, added to the fact that you will have some amount of carbon dioxide dissolved in the urine itself, right? And also, of course, your body can dump protons, H plus, into the urine if your blood is too acidic, right? So remember, H plus makes your blood acidic, pretty much makes everything acidic. So your H plus is the same as protons. So when you remove protons from the body and dump them into the urine and get rid of them, your blood becomes less acidic. But you can also absorb bicarbonates from the urine and send them back to your blood to make your blood more alkaline. And in doing this, of course, it helps you to control the acid-base balance of your blood. Remember, your blood is supposed to be at a pH of 7.35, 7.45. Remember, if it's too alkaline, you get alkalosis, which means the blood doesn't want to give up oxygen, which is a problem. If your blood is too acidic, you get acidosis, which means that your blood does not properly load itself with oxygen, which is another problem, right? So you need to be able to control the pH of your blood, and the kidney helps with that. And of course, it also detoxifies free radicals and drugs. Basically, your urinary system is used to dispose of many poisons we take into our body. And many other drugs that you take are poisons. I'm assuming everybody knows that, right? And this is, of course, why a good test for substance abuse is going to be a urine test. You don't have to do a blood test. You simply test the urine. Because when somebody is on any kind of drug, heroin, crystal meth, all of those things, cocaine, your body doesn't want these things. Right? They're a foreign substance, and your body ends up excreting them, or secreting them, I should say, into the urine, and then the urine, of course, carries them out. Right? But also, even something simple like Panadol, right? Advil, acetaminophen, aspirin. These things also are not substances your body produces, for the most part, or wants. So you'll find them in the urine as well. So let's say somebody takes an overdose of pain medication. This happens a lot, especially if with acetaminophen, right? That's like the Panadol. Panadol is extremely easy to overdose on, right? On the packet says what? 2 Panadols, every 4 hours or 6 hours or something like that, right? People have been known to die by taking 6 Panadol at once, right? As easy as it is, it's easy to swallow 6 tablets, and that ends up damaging the liver severely, and then they die, right? Now, I'm not saying everybody's like that. Obviously, there are factors that will determine how bad it is, but Panadol is extremely dangerous when you take too much of it. So it is good that you can remove the Panadol as quickly as you do. And of course, if somebody dies of poisoning or something, you can extract urine from their bladder and do a toxicology test on it, and it gives you an idea as to what might have poisoned it, right? So it's all good, right? All right, that's it. In terms of waste, we consider the urinary system primed for the removal of what we call nitrogenous waste, right? I'm assuming everybody remembers nitrogenous waste. So there are 3 main types of nitrogenous waste. You all know urea. This is why it's called urine when you urinate, urine, all of that is the urea in it. And the urea is produced by the deamination of your amino acids. So here's the thing. I'm sure I'm about to tell you something you know already, but let's make sure. Your body does not store protein the way it stores fats or the way it stores carbohydrates for whatever reason. We won't get into the reason, right? It has to do with the fact there are too many different types of protein, but still. So here's what your body does. When it has excess proteins, it breaks the proteins down to amino acids, of course, and then it converts these amino acids to acetaldehyde, which is used to make ATP. During the process of making the acetaldehyde, you produce ammonia, right? Because you have to deaminate the amino acids. So you have to take off the amino group, which is NH2. And the NH2, of course, in water, you get NH4, which is ammonium, right? And this stuff, as you know, is a cleaning agent for a reason. It is extremely poisonous. So your body can't have ammonium floating around in there because it will simply poison you to death. So what you do is you have to take the ammonium, and besides, ammonium is just ammonia dissolved in water, right? And you have to convert that ammonium to urea, which is how you get this, right? And to make urea, you need carbon dioxide in the process. So making urea,
the jail candidate once, and he was replaced with Venetia Phillips. So he decided.
okay. Anyway, so that is how now you use carbon dioxide and the ammonia to produce urea. we don't need to get into the specifics of it, right? We're not going to go into the chemistry of it. Just be aware. Ammonium, which your body makes when it breaks down amino acids, is added to carbon dioxide. There's water involved, but we won't get into that. To give you urea. Why do you do this? Because ammonium or ammonia, whichever one you want to use, is extremely toxic. Whereas urea is less toxic, right? Remember, no, it's not non-toxic, it is less toxic. So ammonia, if for example, your body did not do this, within a few days, you could very, very easily get poisoned by ammonia. So if your body was just making ammonia and just dumping it into the bloodstream, in a couple of days, you're dead, right? Urea, even if your kidneys stop excreting urea tomorrow, normally you have anywhere from 2 weeks to 6 weeks, depending on your diet and the person before you die. It's a lot less toxic, right? Now, another form of nitrogenous waste that you may have or may not have heard of is uric acid. Human beings produce a tiny amount of uric acid, right? Now, here's the problem with uric acid. Uric acid tends to form crystals very easily, right? So if your body makes a lot of uric acid and moves that uric acid into the bloodstream, you end up over time getting uric acid crystals blocking up your blood vessels, which is not nice. Blocking up your kidney, which is not nice, right? So your body really does not do very much with uric acid. Small amounts, too small to really cause any problem. As a matter of fact, normally mammals in general, like ourselves, don't play around with uric acid. Uric acid is something you normally find in great concentrations in birds and in lizards. You know when they say lizard poop and bird poop, you have this whitish stuff in there. The white part of it is going to be the uric acid, right? It just so happens that it takes a lot less water to produce uric acid than it does urea. Urea needs more water. So because of that, birds and reptiles can do better in low water environments than mammals, which is, in case you're wondering, the reason, one of the main reasons why in deserts you find a lot of lizards and birds and snakes and all of these other things. You don't find a lot of mammals because mammals cannot do the uric acid thing, right? All right, continue. So that's the second type of nitrogenous waste. The last one you're likely to come across is this one here, creatinine. Now, if any of you know anything about people who do a lot of weightlifting, bodybuilding, that kind of thing, creatinine is something that bodybuilders take in large quantities because creatinine helps with recovery from exercise. How does it do that? Your body produces something called creatinine phosphate, right? The creatinine phosphate is used by your muscles to make ATP rapidly, okay? Virtually instantaneously. So when you're exercising, trying to lift heavier things, and your muscles are running out of ATP, you have creatinine phosphate stored around the muscles. Karen, your camera's on. So what happens? You just break down the creatinine phosphate real quick, and you get the ATP almost instantaneously, right? But when you break it down, you're left with excess creatinine, and some of that gets excreted, right? So you won't remake all of it instantaneously, which is the reason, of course, why these guys will be taking the creatine powder to make up for what they've lost, right? And actually, it does help with recovery, right? Lots of professional athletes, they can tell you that when they do their training and their heavy exercise, they take creatinine powder, and then a day later, they can go as hard, almost as well, 2 days realistically. They can go almost as hard as they did, right, before. Otherwise, it can take days to recover the creatine phosphate you use during exercise, right? So remember, this excess creatinine being lost is essentially as a result of exercise. You're breaking down the creatine phosphate, you're giving off the creatinine that passes through your urinary system, and you lose it. So you need to get back more, hence you take the powder, right? All right. So those are our three main types of nitrogenous waste. All right. Now, if you do happen to be one of the unlucky folks who will experience renal failure, and statistically consider the number of people in here, about 2 of you will undergo this at some point in your life. Here are some of the forms of here are some of the forms of malfunction that you'll come across, renal failure. These are the symptoms, right? First of all, you have something called azotemia. In azotemia, you find excess nitrogenous waste in the blood. Obviously, there's always going to be a small amount of nitrogenous waste in the blood, right? Because you're continually removing it as you go along, right? But if you start to get a buildup of this waste in your bloodstream, you will get azotemia. Azotemia, no surprise, is usually a sign of your kidneys starting to fail, right? Now, if the azotemia gets bad enough, you end up now with uremia. Right? So remember, azotemia, excess nitrogenous waste starting to build up in your blood. You might not be sick at this point, right? But you get in there. When you actually start to feel the muscle pain and the tiredness and the nausea and the vomiting, that is when now you're starting to get the toxic effects as the waste starts to accumulate in your body. And that leads you now to uremia. Right? So azotemia, untreated, leads, of course, to uremia. Right? All right. So what does excretion mean? Basically, excretion, as I'm sure y'all remember, because I think we did excretion versus digestion already. Excretion is the removal of metabolic waste from the body. It can't just be any waste. It must be metabolic. Right? So that metabolic waste is something that has to be created by the body. Sorry about that. Anyway, remember that it's the removal of metabolic waste. Okay? So what are the types of metabolic waste? Remember, we should have mentioned these already, so you all should know, right? So, carbon dioxide, we produce carbon dioxide during respiration, hence it must be metabolic waste. We produce a small amount of water as well during the process of aerobic respiration. So, technically speaking, the water in your sweat and some of the water in your urine is also metabolic waste, right? I think on average, human beings produce about 10% of the water that we actually need on a day-to-day basis. 10% is not a whole lot, obviously, but I mean, it's something, right? Now, urea, of course, water, lactic acid, all of these things are going to end up in the sweat, right? So the integumentary system, when you're sweating, your sweat is a form of excretion because most of the components of it could potentially have been produced by your body, right? In the digestive system, once again, you see how often water shows up? Because we make water, right? Small amounts, but still. Some of the salts, we're talking about like bile salts now. right? So when you produce bile and you dump bile into your digestive tract and the bile leaves your body, that is excretion. So I know a lot of you at high school, you were told that, you're told that defecation is egestion, while urination is excretion. Is not true, right? The digestive system involves both egestion and excretion. And remember, egestion is the removal of non-metabolic waste, things that your body has not messed with, right? Now, hopefully, the salts are in the bile, so it make sense that the bile is here as well, right? All right. So, hopefully, all now remember what excretion is, right? Now, let's talk about the kidney itself, because obviously the kidney is the major organ of the urinary system, right? Now, it says here the kidney is retroperitoneal. I am hoping that even without knowing what this word is about, that you can suss out the meaning, right? For example, something retro, like retroactive or retrograde, is behind, right? So even when it's people talk about retro fashions, they mean fashions from long ago. So it's from behind in the past, right? So it's behind something. And peritoneal, I'm hoping that once you see this word, the term peritoneum comes to mind. And what is the peritoneum? It's the serous lining of your abdominal cavity, right? So I'm hoping just seeing the word, you can figure out that it is the kidneys are behind the peritoneum, hence retroperitoneal, right? All right. It also says here that they're at the level of T12 to L3. Now, I should not have to tell you what T12 and L3 are, right? If I need to, please let me know, but you should know what they are. If you don't, try to make sure you ask me, right? Anyway, because each kidney here is about 160 grams and approximately the size of a bar of soap. Now, one thing that we'll be doing this week is that we'll be dissecting kidneys in the class, right? A quick sidebar. In the class this week, we're doing two things. We're doing respiratory physiology and we are doing kidney dissection. Now, as you know already, the numbers in the class are ridiculous, right? So here's what I'm going to try and do. I am going to try to have, I brought some, I'll be bringing some apparatus with me that can be used outside of the classroom. So as long as we have like an empty class, I can send a half of you to the one thing, keep the other half in the lab. And then after like an hour and a half or so, I can swap you around, right? So that we don't have everybody tripping over everybody in the class at the same time. So we'll see how that works out, right? So keep that in mind. But remember also, there was plenty of room in the 8 o'clock. Well, we normally start 8:30. There was plenty of room in the 8:30 session, right? By plenty of room, I mean like about 6 spaces. All right. Let me go back to what we're talking about now. So anyway, so it says here now the shape of the kidney. Now, I have had this happen to me in the past where you've given the students a question and asked them to describe the kidney. And on more than one occasion, I've seen them write the kidneys are kidney bean shape. That don't make no sense. You can't write that something's the shape of something, which is the same as using the same term as something. That don't make any sense, right? You cannot say the kidneys are kidney bean shaped. You have to say it this way. The kidney possesses a lateral surface convex curvature and a medial surface concave curvature. Or simply put, it's laterally convex medially concave, which kind of describes a banana as well, but at least it gets the curve in there, right? Then in addition to saying that the kidney is laterally convex and medially concave, you can also mention the coverings, okay? So you have the renal fascia. And the renal fascia is important because it holds the kidney to the wall of the abdomen. Because, of course, the last thing you want is for your kidneys to go bouncing around the place when you're running around, right? So the renal fascia helps to paste or tape the kidneys in place. If you want to think of it that way. If this one is not on Moodle as that is put up, I thought it was, but that could be wrong, right? So if it's not there, have no fear. It will be right after this. But actually it's there, but I don't think I have Mobi underneath it. Yeah, don't worry about it. It'll be there. Now, I'm almost tempted to stick a pin and put it up there right now, but it's probably not necessary. Or do you want me to do that? You want to follow along with your own, so you can make notes on it? Is that what's going on? Should I take a minute and go do it now? All right, not too important. Cool. All right, so remember now, you have the fascia that is helping to stick they are taping away because it's like a layer, the kidneys to your abdominal walls. Why? Because when you're moving around, the last thing you want is for your kidneys to go moving around as well. There are tubes coming out of it, they'll get twisted up, right? You also have surrounding the kidney an adipose capsule. Adipose, as you know, is fat, right? Now, the fat is there to cushion the kidneys. So when you drop or something, of course, you know your internal organs are smushing to one another when you trip and drop. So it's good that that you have this cushion around all of them, this fat layer that helps to absorb the impact. So it is very unlikely that somebody would trip and drop, hit the ground, and one of the internal organs bursts. It would be kind of a lousy setup if that was the case, right? So you have this cushion of fat around them, right? And then on the surface of the kidney itself, you have what is called the renal capsule. And the renal capsule is made up primarily of collagen fibers. Now, I don't know if anybody in here eats kidneys, right? It's not really my thing, and I don't judge. But if you've ever prepared kidneys, as in kidneys that come from the supermarket or the butcher or wherever, more often the ones that haven't been cut up, of course, more often than not, you'd have to peel off a layer on the surface of the kidneys. And the layer is sort of translucent and it's tough. If you leave it on there, and you'll find the same thing on liver as well. If you leave it on there, the thing is hard to eat, right? That there is your renal capsule. And it is there obviously because I mean when you try to eat it, it's very tough. It's going to provide strength and increase the damage resistance to the organs you'll find these capsules on, right? So we have three major covering layers. We have the fascia, outermost layer, the adipose layer underneath it, then the renal capsule right on the surface of the kidney, right? So here we have a kidney. Now, this is now looking at the internals of the kidney. And this is something you guys should have seen at CSEC level, right? So you should already know the major layers. If I say cortex, first of all, anytime you see the word cortex, you should realize that this is the outer part of something, right? Cortex is always outer. So cortex will always be the outside bit, and the medulla will always be beneath the cortex, right? So cortex, outermost part, medulla, below the cortex. It's not going to necessarily be the innermost part, but it will simply be below or within the cortex. So we have cortex and we have medulla here, right? And we also have an area here called the renal sinus and the hilum. Now, as I said to you, I am assuming you all recognize these things from CSEC, right? I'm assuming you also recognize the minor calyces and the major calyces and all of these things. I'll be going through them in a little bit of detail though, but just have a look at this. See if there's, obviously this is something you could be asked to label in the future. Refresh your memory here. This is of course the renal artery and the renal vein, right? I'm sure the color helps with that, right? And as we're going along, we'll make sure that you refresh your memory, right? Now it mentions here the cortex is about 1 centimeter thick, right? And the cortex will tend to contain the nephrons. And the word nephron is a word that you should know, right? The nephron is what we call the functional part of the kidney, or it is the basic unit of the kidney. And the basic unit, remember, is the smallest part of the organ that can do the major functions of the organ, right? So the nephrons, which we're about to look at, basic units, right? And most of them are found in the cortex, right? At least the bulk of the nephron will be found in the cortex. The medulla now is going to contain several important things, right? The medulla is going to contain collecting ducts as well as some special nephrons, which we'll talk about in a little bit, right? So this here is looking at the setup. If you look here, this is one nephron. I'm sure you all recognize all the squiggly lines from the nephron. And then you will see the nephron itself, they attach to these structures here, and these are your collecting ducts, right? So here's the basic idea. The nephron takes blood in, filters out the excess urea and water and salts and all sorts of other things, and then it passes them through the tubules that make itself up. And during the passage of that fluid that you get from the blood, the filtrate, as we call it, substances are reabsorbed and substances are secreted. And by the time that fluid gets now to the collecting duct, that fluid is now urine. And the nephron then dumps the urine into the collecting duct. As you can see here, multiple nephrons will connect to the same collecting duct, right? Now, I also want to show you this weird looking nephron here. See how long this thing is? Most nephrons are like this one here, right? It's a cortical nephron because, of course, it's mostly in the cortex, right? So keep that in mind. Cortical. But a small percentage, about 15% of nephrons are like this one here. And this one here has a lovely name. It's juxta medullary nephron. Right? Juxta medullary nephron. Now, as usual, most of these scientific terms, if you recognize the parts of it, it can tell you what it is, right? So, for example, juxta is like juxtapose, right? I don't know if you all know what juxtapose means. It means you take two things and bring them close to each other, almost until they can overlap so you can look at them. So you're juxtaposing them. You're moving them into the same space, right? Then you have medullary. Obviously, that means medulla. I mean, what else would it mean, right? So juxta medullary nephron it means that you're bringing some part of the nephron close to something else. We don't know what that is yet, but something's going on there. But the medullary tells us that the bulk of it is going to be in the medulla. Just the way the cortical nephron, the bulk of it is going to be in the cortex, right? So keep that in mind. I'm going to mention that in greater detail in a little while, right? Then now, I want you also to notice that these nephrons are all connected to collecting ducts, which means all the urine that they produce run down the collecting ducts. And there's a special area inside your medulla where all of this is happening, right? If you notice here, this has sort of a rectangle, well, here a rectangle, a triangular shape right here. You can see the color is different too. That's because what you're looking at there is called the renal pyramid, okay? So this area right here is where you're going to find this, okay? So we have our cortical nephrons and our juxtamedullary nephrons. They're dumping their urine into the collecting ducts. The collecting ducts are running down into the renal pyramids, which is that special part of the medulla for them, right? They must be dumping the urine somewhere, right? At the bottom of the renal pyramid, you have this area called the renal papilla. And that connects to a tube called the minor calyx. And you can see little holes here in the renal papilla. So the urine from the collecting ducts leaks out and goes into the minor calyx. Melissa, I'm seeing you. Right? So keep that in mind, right? So, all right, let's go one more time. All right, let's go one more time. If I can find my space. All right. Let me clean it up, and then we're going to go again from scratch, right? So remember now, two types of nephrons. We have the cortical nephron, called thus because the bulk of it is in the cortex. We have the juxtamedullary nephrons because it has this massive part of it is down in the medulla, right? They have slightly different functions, which we'll talk about later. Don't worry about that right now. But all the nephrons end in your collecting duct. Multiple nephrons are dumping urine into the same collecting duct, right? And you find the collecting ducts in these special areas of the medulla called the renal pyramid. All right? Pyramid because it's kind of shaped like a triangle. Actually, it's more like a cone, but we call it a pyramid, right? So now, at the end of the renal pyramid, you're going to find some little holes where your collecting ducts empty out, okay? The area where you have the holes is called the renal papilla. And that area also connects with a tube called the minor calyx, right? So you make the urine in the nephron, pass the urine to your collecting duct, right? That urine runs down through the renal pyramid, empties at the renal papilla into your minor calyx. And yes, I'll be putting up this recording. This is an actual human kidney. Obviously, this one is preserved, so some of the colors are not so easy to spot. It's a little hard to tell, but at least you have it labeled here. I mean, I won't give anything like this because it's too hard to interpret. Can you imagine this being printed in black and white? I mean, asking you to label this? That would be a hell. So I wouldn't. So don't even say too much about this one. All right. Now, here's what I want to do. No, I'm going to do a little diagram here to show the progression of urine through your nephron. Okay. I'm going to be very simple, very basic. It's just to refresh your memory about how this is supposed to go, right? So please keep that in mind. I'm just refreshing memory right now. Don't so everything you're seeing here that I'm about to do, I am hoping and praying that you remember from CSEC, right? It will make life a lot easier. So let's start with a basic drawing of your nephron. Remember, very basic. So I'm doing a CSEC level drawing. Now, as I'm going, y'all hopefully are following along and saying, oh, I know what that part is. I know what that part is, right? All right. Can't get much simpler than that, right? Let's add something important here, which I'm hoping you all know the name of. All right. So let's work our way through this. This, of course, is a blood vessel here. We call this blood vessel the afferent arteriole. And on the other side, we call this one here the efferent arteriole. All right. So now blood flows through this afferent arteriole and then moves into this structure here. And this is a name that most of you will remember, which is the glomerulus. Right? Now the space the glomerulus is sitting in is called the capsule space. Right? Let's call it capsule space because this structure right here is the Bowman's capsule. All right, as I said, hopefully everything I'm saying so far should sound familiar to you, right? Now, as blood passes through the Bowman's capsule, as blood passes the afferent arteriole, it enters the Bowman's capsule. the glomerulus in the Bowman's capsule. And then it leaves via your efferent arteriole. Now, here's the thing. I want you to notice that the afferent arteriole is bigger than the efferent arteriole, right? Also, hopefully you remember that the glomerulus itself is a network of capillaries, okay? So it's a network of capillaries, fenestrated capillaries. So you have a big tube going into the glomerulus with all the capillaries in there, and a small tube, which is the efferent arteriole, coming out, right? So if a big tube goes in and a small tube goes out, there's going to be a buildup of pressure because the blood is pushing into the glomerulus and it is not leaving at the same rate because the tube going out is smaller than the tube going in. As pressure builds up inside the glomerulus, because the glomerulus are capillaries, substances that are small enough can leak out, right, into the Bowman's capsule space itself, right, because of the pressure. There's so much pressure in the glomerulus. Because afferent is bigger than efferent, you squeeze now fluid containing substances out of the glomerulus, right? That eases the pressure and makes it easier for blood to leave at the efferent arteriole, right? All right. Now, this part here is the proximal convoluted tubule, or the PCT, right? You also remember this one too, right? Proximal convoluted tubule. It's called proximal because it's the closest one to the Bowman's capsule. It's called convoluted because it twists up, right? So convoluted just means twisted up. So now when you squeeze substances out of the glomerulus, these substances go into the capsule space and then drain into your proximal convoluted tubule, right? And as the substances pass through the proximal convoluted tubule, you will have things being taken out of them or reabsorbed, right? So you're talking about water, for example, will get reabsorbed here. Amino acids, glucose, of course, is another big one. Some salts, and even some hormones. Right? So these substances will be pulled out of, we can call it the filtrate, since it got filtered out of the blood, right? So the stuff coming out of the glomerulus, we can call it the filtrate. That filtrate goes to the Bowman's capsule, then goes into your proximal convoluted tubule. Once it gets there, some substances are recovered. Not everything that comes out of the blood you want to get rid of. So we say that substances are reabsorbed then by the blood, right? So hormones and water and amino acids and glucose and salts, all of these things end up going back into the blood, right? As the filtrate travels down the PCT. So as the filtrate is moving down the PCT, the volume of it is dropping because you're pulling things out of it, right? And some of the things you pull out of it, you don't even want to pull out of it. So for example, urea, about 50% of it, roughly half of it goes back to the blood. So you're going through all of this trouble to get rid of the urea, and only half of it you get rid of, right? Because urea can pass through cell membranes very easily. And it happily diffuses from the filtrate right back into the blood. So obviously, you can't lose more than 50% of its diffusion, right? So now, by the time now you move down the PCT, you get down now to this other portion here, which I'm sure this is another one that you all tend to remember, is the loop of Henle. Right? Loop of Henle. Now the loop has a descending part, which obviously goes down. Right? So it's descending. And it has an ascending part. Right? Now, remember, I'm just telling you the stuff you learn at CSEC. I'm not going to get more complicated at for now. As the filtrate passes down the loop of Henle, down the descending part, of course, water gets pulled out of it. Right? So you pull out some water in the PCT, you then pull out more water in the descending part of the loop of Henle. Right? Then now, as that filtrate makes its way now up the loop of Henle, instead of pulling out water, you know, we pull out salts. Right? So the water stays this time, and now it's the salts that come out. So, descending loop of Henle, the filtrate loses water, so the volume of the filtrate goes down, and the concentration of the filtrate increases, right? Because if you're losing water, but you're not losing salt, something must get more concentrated, right? So it must be getting saltier and saltier and saltier as the filtrate flows down the ascending loop of Henle. When the filtrate then turns now and goes up now, the ascending loop of Henle, the filtrate is going to start losing salt, but it's going to keep the water. And if it's losing salt and keeping water, it means the concentration now starts to go down instead of up. So it gets less and less and less salty on its way up, right? Until its salinity can be even less than that of blood, right? Which is where it's coming from to begin with. But we won't worry about that right now. Then now, the filtrate moves into this part here called the DCT, distal convoluted tubule, right? And that filtrate now just lost plenty of salt and water, goes into the DCT. So what happens here? You still lose some water at the DCT, right? But mainly what is going on at the DCT is secretion, right? And secretion in two directions, in and out. Now, remember, secretion now is an active process where you're moving substances in whichever direction you're trying to get them to go in, right? So basically, here's why you need secretion. Some substances in the body, in the filtrate, you still want them, right? Some hormones, for example, you still want them, but they're in very, very low concentrations. So the only way to get them back in the body is basically to grab onto them and push them out of the filtrate. So that is active transport, right? But also, some substances from the blood supply, you don't want them in the bloodstream. And because they are in small, low concentration, as we said before, it's hard to get rid of them. So you have to push them out of the bloodstream and into your filtrate, which is once again secretion. If you need ATP to move something, a substance in any direction, that's active transport, and you're secreting it. Okay? All right. So for example, we mentioned drugs earlier, right? So the marijuana, I don't know if you all know, I assume everybody knows by now, that the active ingredient, for example, in marijuana, well, one of them is THC, which is tetrahydrocannabinol. Can't remember the name of the other one right now. But when you smoke the ganj, as I'm sure some of you are want to do, the THC gets pushed into the urine at the distal convoluted tubule, right? So it lowers it up in there, which of course is why you can do a drug test and get a positive result for THC from the urine, right? I remember this story a couple years ago. This is America, where you had people, American football, which is not really football because they don't really kick the ball more than so. But still, whatever, it's their sport. You had these players, and they are regularly drug tested, right? And you had this one guy apparently who took a weekend with his friends and went to Las Vegas or wherever it was and smoked and drank and, did the Las Vegas thing and had to do a drug test almost about a week later. And he knew he was going to fail the drug test out hell. So when he got home, he went to his girlfriend and borrowed some urine from his girlfriend and stuck it in to the exam. And he basically, because I can't watch you pee, dignity, right? So he went in the bathroom and he had a little bag strapped to his leg. And he pulled out the bag and he poured the girlfriend's urine into the test cup. And then he brought it back and said, here's the doctor. Here's my urine. And he had to sign a form and say, this is his urine and blah, blah, blah. It hasn't been adulterated, whatever, whatever, right? And then a week later, yes, somebody knows the story. A week later, he got a response. He says, he's completely drug-free, but of course, he's also pregnant, right? So Daniel knows the story. So yeah. Also, by the way, weirdly enough, there are some places there's actually trade in urine, especially amongst people who work for the government, the US government. It's actually based on a real story. So the US government, for example, if you're working in certain areas of the US government, security issue, if you're on drugs, so you mustn't test positive for any drug at all. And these guys now they actually go and buy urine from people to test. Yes, Talia.
Sir, in regards to what you just said, I'm not sure if it's true. I just want to confirm, but I saw that in a movie, and it turns out that it would, if the guy tested positive for like being pregnant, that would mean he has cancer.
If it's okay, if you have prostate cancer, it can increase the level of something called HCG, which is human chorionic growth hormone. And if you took a pregnancy test as a male and it comes back positive, it normally means that you have high HCG levels, because HCG is what the pregnancy test is testing for. In females, you get it because, of course, you're pregnant. In males, you get it because of overproduction, usually due to cancer. So you're quite correct. So if a male, for example, has swollen testicles and it hasn't gone down, maybe he's peeing a little blood, and he's concerned, he can go buy one pregnancy test and test. And if it's positive, he knows he has cancer. If it's negative, he has an infection, right? But keeping in mind, of course, that normally when it's positive, it's because the cancer gone a fair bit, right? Yes, Juvin.
Sir, I heard somebody say, like, if you drink tomato juice.
No, it doesn't work.
Okay, all right.
I'm gonna be 100%. None of the things if you go online, they must sell you all kind of thing. As if you're mixing the urine with fool test, not go so. Right? Not go so. I don't understand the gas comment testing.
Sir, you said the balls, if it swell up, it could be gas.
How the hell gas get in this?
Sir,
I know
Sir,
I know we call them balls, but they're not footballs there. Right? You're not going to find a place to insert a pump. Right? Trust me, I've checked. Right? So keep in mind, as I said, anyway, Abby, yes, it is possible for a male to test positive on a pregnancy test, but it is always bad. Right? Always bad. Right? It's never a happy occasion where a man gets a positive test for a pregnancy test. Anyway, so after secretion, of course, the urine, we can call it urine at this stage, moves into this last structure here, and this is your collecting duct. And from there, the urine makes its way out of the body, right? Now, I'm hoping everybody remembers this stuff from CSEC. All right, at the very least, I'm hoping this refresher helps to remind you. Because we're going to be going forward, and of course, we're going to be adding to this story here, right? This is the basic story. Now it's time to add more to it, right? By the way, y'all remember our lab quiz coming up Tuesday, right? Wednesday, right? I have one to give back to you. And then you're supposed to be doing a combo lymphatic and cardiovascular lab quiz. So please don't forget, right?
and it's just on heart and lymphatic system, right, sir?
I mean, if you want it, I'm more than that. I can facilitate if you want.
No, that's fine. Thank you.
Never let it be said that I'm not willing to help. If you feel that the test too easy, I can always add to it.
No, sir, that's fine.
Anyway, because I know I'll mash up the last one. So I need something more challenging in your life. Anyway, this here now. What happened to the video? I see something about the video you're not seeing. I'm confused. All right, I just got a message in the chat that said the videos. Didn't I upload? Oh, are you talking about the previous videos? I thought I uploaded the ones I had already. I need to go and check. If not, I'll upload whatever is left over, but it should be most of them. But remember, obviously, the Tuesdays I was down there, there's no video for those, clearly, right? All right, anyway. The next thing we need to look at is the path that blood takes through the kidney. Now, this is something that you probably would not have done at CSEC, right? But it's going to be something that you're going to need to know. You're going to need to be able to trace the flow of blood through the kidney, right? So that is what this lovely little flow chart here is close enough to a flow chart, right? So you notice here, we go from renal artery, because the renal artery branches off of the aortic, the abdominal aorta, as you know, and then that goes directly into the kidney at the area called the hilum. And this is the second time you should have heard the term hilum, right? The first time when we did respiration, hilum was the area where the tubes went in and out. For the kidney, you buck up on hilum again, and it is still the place where the tubes go in and out, right? So entering at the hilum, your renal artery. And then from there, the renal artery goes and splits into interlobar arteries. So that means, of course, that your kidneys have lobes, right? Once again, if a part of the kidney get damaged, the rest of the kidney should be able to carry on. Right? You'll notice this lobular arrangement is very common in the body because of that, right? Then now from the interlobar arteries, which as I said, they are run between the renal columns, which I'll explain what that is in a minute. Between the lobes, you get the arcuate arteries, which run above the renal pyramids. Then the interlobar arteries, which run in the cortex. Then finally, the one you know, the afferent arterioles, which of course go to the glomerulus, and then to the efferent arterioles, right? And from the efferent arterioles, it goes to these things called the peritubular capillaries, which are going to be very important when I talk about reabsorption and PCT. And then from there now, once you pass the capillaries, of course, you go into venules and veins normally, right? So in this case here, you go to interlobular veins, and the arcuate veins, then interlobar veins, and finally, the blood leaves in the renal vein, right? You're going to need to know this sequence, right? So here we go. Renal, let's do green. So renal artery, blood comes in, splits up to go into your interlobular arteries, which run between the lobes, right? So in between here, so I'm going on the other side, in between here, so instead of running beside the renal pyramid, then they go up and over, like that. The part that goes over is called the arcuate artery, right? Then when it goes from up to come back down, the down part now is your, sorry. Sorry, I almost sneezed. Anyway, where was I? Yeah, so so that's interlobar. Oh, I should mention this here. From the arcuate artery, it spreads out now into these tinier blood vessels up here, which are your interlobular arteries up here, right? So you can see them here again. And remember, these are the ones that are going to go to your afferent arterioles, right? So those little tiny ones. So the little tiny ones up here, the interlobular arteries, are going to go to your afferent arteriole, glomerulus, all of that, and then eventually come back and end up now in your veins. And of course, you're going to reverse the direction to leave. So interlobar artery, interlobular artery, interlobar artery, arcuate artery, and then renal vein, and then they come out, right? So if I give you a diagram like this to label, or if I ask you in a short answer question to trace the flow of blood through the kidneys, and that sounds like a nice question, you should be able to draw me a flow chart or do something like this, right? Either one would work. So keep that in mind, right? Now, that said, let's talk about the renal corpuscle. Now, once again, I hope you all remember this from CSEC, but let's just make sure you know what the renal corpuscle is. The renal corpuscle is simply the combination of the Bowman's capsule, right? That's one. The capsule space, that's two. And the glomerulus itself, that's three, right? That is your renal corpuscle. And the reason why it's named separately is this. This is where you're going to have the process of filtration occurring. Right? And because it is filtration under pressure, we normally call it ultra filtration. So ultrafiltration, filtration under high pressure. Remember how the high pressure comes in. The afferent arteriole is bigger than the efferent arteriole, right? The blood flows down into the afferent arteriole and moves into the glomerulus, right? Which are, of course, capillaries. Now, because the tube going out is smaller than the tube coming in, when the blood makes its way around to leave, there's a smaller opening here and a bigger opening going in that causes an increase in pressure inside the glomerulus, right? Because more blood is trying to squeeze in than the blood that is leaving. And as the pressure builds up now, you're going to have, of course, the leaking, the filtration. You're squeezing substances out of the blood because of that buildup of pressure, right? The pressure is so great that it really should burst the capillaries. Because capillaries are not really designed to take such high pressure. But the capillaries in your kidneys are special. They're surrounded by special cells called podocytes, right? And these special cells wrap around the glomerulus. And because they wrap around the glomerulus, they help essentially to hold everything together. So even under high pressure, the glomerulus, the capillaries, generally speaking, don't burst. Well, under normal high pressure anyway. Obviously, if you have high blood pressure, that's a different story entirely, right? But we'll talk about that later. So you remember capillaries under high pressure in the glomerulus. Why? Because you have a big afferent arteriole going in and a small afferent arteriole going out. So you have blood forcing its way in a bigger tube and trying to leave through a smaller tube. Pressure builds up. And as the pressure builds up, because these are capillaries and they have little holes in them, things squeeze out of the blood. Right? But these are fenestrated capillaries, which means the holes are tiny. So blood cells can't squeeze out of the blood. Big proteins can't squeeze out of the blood. Only tiny stuff, right?
Excuse me, sir.
Yes.
What you had said the capillaries are surrounded by?
Podocytes, right here.
The
They
podocytes.
wrap around the capillaries of the glomerulus to try and keep them together so they don't burst. All right. So now let's do nephron in a little bit more detail, okay? Pretty much everything I said before, of course, is going to still hold. I'm just adding to it slightly, right? So once you filtered substances out at the glomerulus, remember they pass into the Bowman's capsule space, and then from there filter their way down into your PCT, your proximal convoluted tubule, right? No, notice here, it says the PCT is the longest and most coiled of the tubes, and it is made up of simple cuboidal tissue with a brush border. Remember simple cuboidal from EMP1. Simple cuboidal tissue, one layer of approximately cube-shaped cells, which means that they're just about as long as they are wide. They have a large nucleus, normally in the center, and they are great at absorption and secretion, right? And of course, a brush border increases the surface area for absorption and secretion. So they're designed for what they're about to do, which is take things out of the filtrate, right? All right. Then now, of course, below the PCT, you have the nephron loop, or the loop of Henle, as we know it, right? And the loop of Henle has thick segments and thin segments, in addition to having descending parts and ascending parts. Abby indicates for active transport. She's right. The thick segments here are going to be for active transport. The thick segments are for pumping salt out of the filtrate, right? So so as the filtrate passes through the thick part of the loop of Henle, you actively push out salt. Now, please remember this. When it comes to water, you never actively transport water. There's no place in the body where you're pumping out water, right? Why don't you pump water? Because water is heavy. People don't realize, do you realize how dense water is? Water is a very strange substance, you know. It's very weird. Water is very dense in comparison to most liquids you'll find naturally. And as remember, natural liquids, right? Most natural liquids float in water. I mean, all oil float in water, right? Alcohol float in water if it don't dissolve in it, right? So water is weird because of that, right? And it's incompressible. You can't squeeze it into a smaller shape, right? Most of the liquids, you can kind of squeeze them. So because of that now, it's too much effort. It's too dense. It's too hard to move. You don't want to pump water. If you want water to move, here's what you do. Water loves salt, right? Water always follows salt. So if you want water to go someplace, all you have to do is pump salt now to that place and water runs right behind it, right? So if you want to take water out of filtrate, right? All you need to do is actively transport the salt out and the water will happily follow behind the salt, right? And you don't have to expend any energy moving the water, which, as I said to you, is an aggravation, right? Now, that said, the thin segments of Lubbock Henley tend to be permeable to water. So water will tend to leave the thin segments, right? All right. Then, of course, from the PCT, we get to the distal convoluted tubule. And remember what we said about DCT already? It is involved mainly in secretion. Yes, there's a certain small amount of water being reabsorbed here, but its main function is secretion, right? And the cells of the DCT are cuboidal cells, but they do not really have a brush border per se, right? Now, we're supposed to be looking at slides. We'll see if we can. Yeah, we'll squeeze in the slide time on Wednesday too. So that means on Wednesday you're doing respirometer, a spirometer, which is plenty of breathing exercises. I'm sure you'll manage that, no problem. You're also dissecting the kidney. You have to draw external, internal of that. And then we're trying also to draw the kidney slides. That part, the slide part might have to be done online, depending on how time is. Because, you know, we're trying to squeeze in a lot. And we missed the first 3 weeks, basically, right? But we'll see where it goes, right? All right. Now, the DCT helps to form something very, very important called the juxtaglomerular apparatus. Now, do you remember juxtapose? You're putting two things near one another, so you can look at them, right? They're almost in the same space. But before I had said juxtamedullary nephron. Now I'm saying juxtaglomerular apparatus. So obviously, juxtaglomerular, you're juxtaposing something with the glomerulus. You're putting something near the glomerulus, right? So what are we putting near the glomerulus in this case? The distal convoluted tubule, right? And afferent and efferent arterioles. So all of them now are going to be near each other to form the juxtaglomerular apparatus. And we'll talk about what the function of that is later, right? Which means, of course, chances are that won't be coming on the test. So let's go. Anyway. But be aware of what makes it up, because this is something we did cover, right? Then now, the DCT leads to collecting ducts, as we know. And several of them will join together to form the collecting ducts. So this lovely flow chart here is just to remind you of the flow through the kidneys until excretion, right? So you notice here, we start at the glomerular capsule. Because remember, the capsule is a part of the nephron. Nephron is a part of the kidney. The afferent and efferent arterioles are technically not a part of the nephron, right? They're a part of the blood supply. So it starts at the capsule. Then the filtrate flows into the PCT. Then it goes to the nephron loop, which is the same as the loop of Henle. Then to the DCT. Then to the collecting duct. And remember, from there, it goes to the papillary duct. Then it goes into the tube called the minor calyx. And then the minor calyces merge to form big tubes called major calyces. And then these major calyces will merge to form the biggest tube of all, which is the renal pelvis. And that renal pelvis dumps urine into your ureters. And the ureters are tubes, of course, that leave the kidney and carry the urine all the way to the bladder. And when it's time to urinate, you pass that urine from the bladder to the urethra, where it eventually leaves the body altogether, right? So obviously, this is a lovely little flow chart, and you need to know this one as well, right? Now, this diagram looks extra complicated, right? I know, but there's nothing here that you haven't seen before except maybe down here, which is the vasa recta. And when we talk about the juxtamedullary nephrons, I'll come back to that, right? But this does give you a general idea as to the setup of the cortex and the medulla, right? So keep that in mind. Now, this is, of course, this is a continuation of a simple explanation as to what is going on to the nephron, right? So even if you didn't understand what I said earlier, this is there as well, right? So it goes through once again the major parts and it gives a simple explanation as to what's going through, right? For example, shows the nephron here, shows you all the parts that you need to know how to label. And you do need to know how to label them, right? It then talks about what basically is going on in each. So Bowman's capsule mentions spherical capsule around glomerulus. It should have mentioned that this is the site of ultrafiltration, but we leave it alone for now. You mentioned the PCT here. It says about 75% of the sodium is removed here by active transport, as well as chlorine, and about 67% of the water as well. I'll mention that no. So most of the water, most of the salt, and most of the chloride, shouldn't be chloride, that chlorine is taken up at the PCT. Also remember, most of the glucose, if not all of it, and most of the amino acids, if not all of it, are also taken up at the PCT. The PCT does more reabsorption than any other part of the nephron. This is the most important part for that, right? Even though everybody always talk about loop of Henle, this one still does more water than the loop of Henle, right? And more salt than the loop of Henle too. And loop of Henle does something called the counter-current exchange system, which will not be on test one because I will not have time to go through it. I only have about 10 minutes left. 10 or 12 minutes, whatever, right? It mentions, of course, that we have an ascending portion and a descending portion, right? And remember, the descending portion, if you notice the descending portion, how thin this part of it is, right? And remember, thin part lose water. So water leaves the thin part. The thick parts lose salt. Right? So all of this here is losing water, and these two bits here are losing salt. I thought I heard a comment. I could be wrong.
I'm sorry, sir. I just wanted to query something. You said while salt is being lost, the concentration is higher at the descending
All right.
aspect.
Yeah, thanks for mentioning that. Let me make sure I run through that now. As water is all right. Remember here, as I said to you, you're losing salts to the thick part, right? So you're losing some amount of salts here, right? So like sodium, for example. which means once you first enter the loop of Henley, you will see you lose a small amount of salt because it's not a very big park, right? But you quickly move into the thin segment of the loop of Henley here. And from the thin segment, of course, you're starting to lose water. Remember, right? So water is coming out, but at no point are you losing salt along this way. Only water. All the way down here, all the way come back up. It's just water. Which of course means now that if you're losing water, but you're not losing any more salt, you're only losing salt up here. So right now, the urine or filtrate, whichever one you want to use, is going to be getting more and more concentrated because you have less water and the same amount of salt in it, right? But when this filtrate now makes its way up to this part, you notice that the thick part here of the ascending is much bigger, right? Than it was on the descending. Which means as the filtrate is passing through here, now you're losing salt, but not water. Right? And this is active transport, which means it goes much faster. So you're rapidly losing salt as you're moving up the ascending part, but you're not losing water because it's impermeable to water. So therefore, as the filtrate or urine goes down the loop of Henley, it is becoming more concentrated. And as it goes up the loop of Henley and enters the thick loop, it becomes less concentrated because it's losing that salt, right? All right. I'll have to go back to it again when we do counter-current exchange system, but just remember for now, going down the thin loop, you're losing water. Going up the thick loop, you're losing salt, right? So as it says here, ascending loop of Henley, chlorine ions, active transport out, sodium follows, water does not, because it is impermeable to the movement of water. Water can't flow through the thick part. This mentions counter-current exchange system. As I said, we're not going to do too much of it right now, right? But notice, as it says, the counter-current exchange system sets up a gradient for more salt towards turning loop, less near convoluted loop. But as I said, don't worry so much about that for right now, right? Then distal convoluted tubule here, as it says, you're taking out the sodium chloride, potassium, ammonia, carbonates are removed here. But remember, you also take out a small amount of water. And remember, secretion is happening here as well, right? Collecting duct, as it says, passes parallel to the loop of Henley. And as it goes down, it's saying that the salt concentration is going to increase as well. But we're not going to worry about that right now. We're going to leave that for when we're doing counter current, right? Now, water always wants to move from high concentration to low concentration, right? So in the collecting duct, of course, water also wants to move from high concentration of water to lower concentration of water. Or if you put it the other way around, water moves from a low concentration of salt to a high concentration of salt. But remember, at the end of the day, water loves salt. The more salt is in the area, the more water wants to go there. That's what's going to be important here, right? All right, this little diagram here just gives you an idea as to how your nephrons are set up. And remember, cortical nephrons, 85%. They have short loops of Henley, right? And they get branches from the efferent arterioles, and they get running around them. They get what they call the peritubular capillaries, which are going to reabsorb substances from the PCT. So the peritubular capillary is going to branch off of the efferent arteriole, and it is going to go around the PCT. And when the PCT starts removing substances from the filtrate and dumping it out, they are going to be absorbed now by the peritubular capillaries and carried back to the main blood supply. And that is 85% of your nephrons. The juxtamedullary ones now, remember, are lower down. They're in, right? And they have very, very long loops of Henle. And they're designed to reabsorb water more than anything else. So the cortical nephrons are your main absorbers of everything for the most part, right? So glucose and amino acids and water and salt and everything. The cortical nephrons will do it. The juxtamedullary ones are specialized for water. Those long loops of Henle allow them to absorb more water from your urine, right? That's what they're designed for. And they have a special branch, a special capillary branch called the vasa recta. And this vasa recta is going to allow them to set up what is called a counter current exchange system. Which, as I said, we're not going to really get too much into detail right now because we only have about 4 or 5 minutes left. It doesn't make any sense to do it, right? All right. That said, I think this is just a summary of what I said earlier. This is looking at the glomerulus in a bit more detail. Right? So this is the glomerular capillary itself. And you can see little holes in there. Right? Remember these are fenestrations. And then this is the podocyte now. This weird looking cell that has extended sections of itself that have wrapped itself around the capillaries to help, as I said, stabilize it, right? But it wraps itself around in such a way that it doesn't block the holes too much. So it allows what we call filtration slits. So where you see these little slits here, you're going to have little fenestrations, so you can still squeeze substances out of the blood, right, without blocking their way, right? And of course, that's the podocyte body. All right. All right, last slide, promise. So the fenestrated endothelium, remember now, the capillaries, pores inside their fenestrations. The pores are 70 to 90 nanometers. Remember, a nanometer is 1,000th of a micrometer. A red blood cell is 7 to 9 micrometers. So the pores are way smaller than a red blood cell. There's no way under normal circumstances blood can squeeze out, right? So you're not supposed to find blood in your urine. Now, the endothelium, of course, is a basement membrane that is made up of something called proteoglycan, right? And proteoglycan has a negative charge. And any molecule that is larger than 8 nanometers with a negative charge gets pushed back, right? So they stay in the blood. So big negative molecules can't pass through. Blood plasma is 7% protein. The blood plasma, the protein in it for the most part, is too small to leave. As it says, only about 0.03% of blood protein will end up being filtered out. And most of that will be reabsorbed anyway, right? It also mentions that podocytes have pedicels and they're also negatively charged around the filtration slits. And they allow particles, a little bigger than 3 nanometers to pass. So what are we seeing here? We're seeing where the way things are set up is that not everything can squeeze out of the blood. Even when they're small, if they have the wrong charge, they can be pushed back into the blood, right? So that is the importance of the negative charges. It helps you to maintain or retain small charged proteins in the blood, right? obviously molecules and ions are way smaller than 3 nanometers. So chloride ions and all of these things are way too small. So these fenestrations and pedicels and filtrations are not going to stop those. This is mainly to stop proteins from leaving. So your urine is not supposed to be high in protein, right? All right, I promise I'd stop there, so I'll leave this. So obviously on Friday coming, I will do a little revision for the in-course test. So I hope I assume everybody will turn up for that one. But let me stop here as I promised. Let me see how far we got. All right, we're at 29 of 71. So we're about 40% of the way. All right, no problem. So we'll put about 40% in the test.
Sir,
Right?
which slide you said you stop?
Slide 29. So we'll stop at slide 29. All right. Any questions before we go? Or is all well? Remember, I'll put this up shortly on YouTube, and I'll put up this PowerPoint presentation right now. All right, so like everybody's good.
Sir,
Yes.
the topics are from unit 1 to
Remember
4.
everything up to this. So everything
Okay.
up to this point.
I also wanted to ask about the podocytes.
Sure.
Are they in between the capillaries that are in the glomerulus,
They're wrapped
like
around them.
scaffolding?
They're wrapped
Oh.
around them.
Okay.
So I always tell students, think about a wrist and wrapping your fingers around your wrist. That's how the podocytes are like. So they're there to hold on tight to them so that they don't pop. As well as I said, prevent large negatively charged molecules from leaving. So keep your proteins in the blood where they belong.
Okay.
All right. As we speak, I am trying to get into UTech to upload this thing. All right, any more questions before we go? All right, sounds like we're good to go. Remember, I will see you all on Wednesday then. But all right, do me a favor now. Can somebody, in case I forget, we need to get the kidneys out of the freezer. Anybody have a lab tomorrow? No labs tomorrow? Will anybody be at Barnet Clinic tomorrow?
No, sir, only on Wednesdays and
Well,
Thursdays.
all right. All right, I'm gonna have to call them then. Because we need to have the specimens taken out. I had them taken out like 11 o'clock last week. And they were still cold when I got them on Wednesday, which is all right.
Type of kidney, sir?
Sorry?
What type of kidney?
What kind of who?
Kidney.
The kidney type of kidney. I don't understand the question.
Like pig, cow, like what type?
You have a preference? I don't understand.
I'm just asking, sir.
Generally speaking, when we use organs, we try to use the ones that are as close to human organs as possible, which usually means pig.
Pig? Okay.
As a matter of fact, if you put a pig kidney and a human kidney around side by side, you really can't tell the difference. Right? We do not use human body parts. You need a special license for that. All right, and we'll never have enough unless you know somebody who's willing to volunteer. We're never gonna have enough.
Shamar will donate his kidney.
Not even one. He's kidneys. Okay.
Yes, sir. Yes, both of them.
He's very dedicated. I appreciate that. All right, folks. So I am putting up the PowerPoint presentation as we speak. Remember, it's in the urinary system section. Anyway, any other questions before we go? All right. All right, folks. It's 9 o'clock almost, so it's bedtime. Or go down on the road time. I don't know what you prefer. Either way.
Knock pot, sir. Knock pot.
Knock pot. You're gonna eat now? It's 9 o'clock.
No, sir. For election.
Okay. I am not really a political person, so it don't matter what thing to me. No matter when I power, then I'm getting the same treatment, so I don't care. Anyway, have fun with that. I mean, I assume nowhere in Mobile has caught fire, so it should be okay, right? Right? No going on there, right? I don't have to be concerned about going down there tomorrow.
Yes, sir. I'm under psychological distress. So just nobody give you the quiz.
You know, if you don't get it this week, I'll get it next week along with your in-course test. Wouldn't that be
Alright.
extra psychological
Sir,
distress?
kindly don't listen to Javine. Don't listen to her. Something
I
they go along
mean,
with her.
she does postponing the inevitable. I don't see the point. And I make it worse on top of it.
Alright, sir. Have a good night.
I will try. All right, folks. I'll see you all tomorrow. Well, Wednesday.